and the aquarium tank, the molecular basis of social organization and behavior. This obviously has important scientific consequences, but it also may provide some help to those of us who have never been able to get fish to survive in our aquarium. So, Dr. Hofmann, thank you.
All right. Can you hear me? That's also okay with the camera? The people in cyberspace maybe something?
Yeah, in fact, the one thing that everyone has said, if you walk around, take the mic with you, because then they're losing it.
I'm not very good at multitasking, so you will have to remind me of that. But all right, well, I first want to thank you for inviting me to your conference here. It's of course very exciting to every once in a while get to speak to a different audience and not just scientists who like to talk about a lot of detail in convoluted language. And so that is of course also a challenge in that I have to step back and get out of some of the detail that might not be really that important for getting across the main points that of work that we're doing and what it might mean and what the implications might be. So what I'm going to be doing is I will set up a framework that will basically explore whether a basic idea that I'm going to propose to you has any merit. And the basic idea is, and some people don't like this idea, is that there are, in fact, molecular universals, as you could call them, that are in place across all vertebrates, in fact, across all animals, across all metazoans, that are involved in regulating social behavior. And that some of these molecular universals that are important in processing the salience of a stimulus, say, in a worm, in fact, may act at the molecular level in a very similar way in our own brains. And as you'll see, as I set up this framework, it is in a way an extension of something that has become very successful in the field of evolutionary developmental biology. And then, after that, I'm going to give you a couple of examples from my own work where we're going to focus on social behavior and particular aspects of it. We use different kinds of approaches, but my focus today will be on genomic approaches to see how we can get at the molecular basis of social behavior. So, so this is what some people refer to as the tree of life, but David Hillis, a colleague of mine in my department here at UT, has produced this wonderful phylogeny, so evolutionary relationships between thousands of species that are on there. You cannot even read them. And has put them in this circular fashion. And I think this is very appropriate because it highlights that the image of a tree of life is actually quite misleading, and in fact what we're having here is a tumbleweed of life. I like this much better in that in many ways we're still fairly close together, but also that there are all these little branches and side branches that are in cross connections that you wouldn't see on a normal tree. So let's focus in a little bit here, in this area up here. It looks like this particular laser pointer doesn't work on that surface. Is that possible? And this one is too difficult for me. All right. I have to push it in the back. That is unusual. Okay, so let's focus in here. And this is where you are. Okay, so we're fairly insignificant in the big scheme of things, even though we think very highly of ourselves. So let's explore, since we obviously are very closely tied together with all kinds of other life forms on this planet, let's explore this a little bit further. So molecular biologists, over the last few decades, and then more recently in the genomic era, genome sequencing have taught us quite a few things about evolution in general and evolution in animals in particular. And the first thing, this is cut off. Okay, so this is just a, there must have been some sort of formatting change. I apologize for this. So the first thing that we've learned is that all living systems on our planet share the same basic macromolecules, such as nucleic acids and amino acids, for the storage, transfer, and realization of information. You all know this. Every elementary school child learns this nowadays. What's more recent and what has still not sunk in into the broader public, I think, is, and sequencing many, many different genomes from many different animals has shown us that, is that a remarkably large chunk of protein-coding genes that we have in our genome are in fact shared and conserved, or they're orthologous across all animals and beyond. So we have about 22,000 protein-coding genes in our genome. The C. elegans worm has about 19,000, and most other vertebrates have about 20,000, and flies have 14,000 or so. But overall, there's a huge chunk that we all share. Right? This is a major discovery that people don't really appreciate. It's not only the nucleic acids that make up the DNA in the genome, but the sequences themselves. And a third important point that I want to highlight here is that comparing large chunks of genomes from different animals very closely, aligning them, has allowed us to discover conserved non-coding regions on the genome that do not encode protein sequences, but that in fact, as we now know, are very important for the regulation of gene expression, so the activity of genes, across a wide range of taxonomic groups. So in a different kind of approach called EvoDevo, or evolutionary developmental biology, and you may have heard of this, what has been discovered and awarded with Nobel Prizes and so on is what Sean Carroll at the University of Wisconsin has called deep homology. And so that is this realization that there are a bunch of genes, such as homeobox or homeotic genes that are indicated here, are in fact shared across all animals. And that in a particular, predictable, and fairly well understood manner, these genes during early development create the patterning, the segmental patterning in the embryo that then both here is shown in a fly and in a mouse, two standard model systems, as you know, that then result in the segmental patterning that we see in the adult animal, including where the appendices, the limbs and so on grow, where the mouth parts grow, in the case of the fly, where the wings grow and so on. So there's a remarkable conservation here, and we know that small variations in these Hox genes and how they are being expressed can lead to diversification and the incredible diversity of living life forms that we see out in the world. Okay. So another example that has become quite popular has to do with the evolution of eyes. So if you go across the animal kingdom, eyes have developed multiple times. Apparently, I need to turn this off as well. Okay. I think I'm not going to touch it anymore. So eyes have evolved multiple times throughout animal evolution. Some people claim maybe 22 times, some people claim 18 times. So you can argue about this, but there is no doubt that there have been several convergent events in the course of evolution that have resulted in eyes for vision. However, as it turns out, there are several genes, and the most prominent one is PAX6, a developmental transcription factor, so a gene whose gene product regulates the expression of other genes, that is in fact involved in eye development and important and crucial, necessary for eye development, and in fact in some cases has been shown also sufficient to induce eye development even in places where normally eyes shouldn't grow, across many, many taxa for many, many of these different eyes. So there's again a deep homology in there that indicates that evolution has made use of a molecular toolbox similar to the toolbox that you have at home, where whenever eyes became advantageous, or structures that resemble eyes became advantageous throughout evolution, it seemed very likely, and we can speculate why that is, that this particular transcription factor got recruited over and over again to aid the development of these structures during animal development. So, as Sean Carroll puts it, ancient genes recycled and repurposed control embryonic development in organisms of striking diversity. So that is another very, very important discovery that shows that the basic outline of our body and our organs is in fact very broadly conserved at the molecular level, and that small variations in these genes or the activity of these genes in their spatial and temporal activity can lead to the diversity and can explain much of the diversity we see out in the living world. Okay? So just as John Wallingford before said, if there are any questions, if I'm saying something that doesn't make sense, please feel free to interrupt. I'm certainly used to that. Okay, so, and we now know or have a fairly good idea that the PAX-like gene first appeared very early on in the Metazoan tree, and basically where you see the first eyes here in Cnidarians, it has taken on this role of developmental importance for eye development. Yeah? Because there are many creatures that do not have genes, and if you were, do not have eyes, and if you were to map which animals have eyes onto this tree, you would see that the most parsimonious explanation is that they have evolved repeatedly independently. Also on the right side, you see there are basic differences in how eyes are structured across the animal kingdom. So there are rhabdomeric eyes and there are ciliar eyes. And then if you compare, for example, the camera eye of a squid or an octopus to the camera eye of a human or a vertebrate, they look in fact very, very similar, but are built one from the inside out and the other one from the outside in. So PAX6 is the underlying developmental regulator. And so it is referred to deep homology, and we'll come back to this concept in a moment, because the homology, so the similarity in these molecular characteristics, is shared across all animals and is best explained and most parsimoniously explained as a result of common ancestry, whereas the structures themselves, such as eyes in this case, have evolved multiple times. Now you may ask a question now in that, well, if for several hundred million years a particular lineage doesn't have eyes and PAX6 is so important for eye development, you would expect simply by drift, as we call it, that the gene underlying eye development that is so important for eye development, PAX6, would in fact disappear because it is no longer used. So another important aspect to understanding this pattern is that PAX6, just like many, many other genes, developmental genes, and as we'll see other genes, are important in many different characteristics of the body. So PAX6 not only plays a role in developing eyes, it also plays a role in other development of other structures. And so as a consequence, it is kept around. So again, let's go back to the toolbox metaphor. You have a toolbox at home. Once a year, you reach in there and use a hammer to put a nail in the wall. But the hammer also has other functions, right? So you leave the hammer in there, the hammer doesn't disappear because you don't use it very often. In fact, you may use it fairly often for other things, such as hitting an intruder on the head or something like that, right? Works pretty well. Okay, so all of this, what I've told you so far, what we've learned from molecular biology, from genome sequencing, from these developmental studies, the basic underlying principle there is the comparative approach, meaning that we look across a number of different animal taxa. In fact, we look sometimes at very weird and exotic species in order to identify the commonalities, in order to identify the universals. And so what I'm also doing here is, I'm putting in a plug for studies on weird model systems, especially in a time where in a large funding agency and the public as a whole question more and more why anybody would work on weird animals like some of those that I'm showing here. And in fact, without this kind of work, we wouldn't be where we are in our understanding of basic biology. So, but this approach is very old. It goes back, 2,500 years to Aristotle, who said, ought we, for instance, to begin by discussing each separate species, man, lion, or ox and the like, taking each kind in hand independently of the rest, or ought we rather to deal first with the attributes which they have in common in virtue of some common element of their nature, and proceed from this as a basis for the consideration of them separately. So the idea has been around for a very long time, and biologists certainly have been comparative for a very long time, but in this modern biomedical environment, this is often forgotten, where some people claim everything we need to know can be done by genome-wide association studies, or maybe by studies of mouse and rat. Okay, so but if we use the comparative approach, it is fairly easy to fall into some traps. And I want to point out 3 of those traps that people fall into regularly if they're not careful and if they don't have a fairly good evolutionary background. The first one, and I'm going to explain this a little further in a moment, the first one is that model systems may be so derived, evolutionarily speaking, so specialized, such as a highly inbred lab strain of mouse, for example, in fact, to mask general principles, and you may not see them anymore. Also, we cannot assume that a particular trait of interest, characteristic of the animal that we might be interested in, or a gene sequence is adaptive in the evolutionary sense of increasing fitness, only because it is there. Right? This is a fallacy that many people commit. And finally, the extent of shared evolutionary history varies across species, and this confounds comparison. It's like comparing apples and oranges. If I have, and we'll go into this in a moment, if I have species that are of different evolutionary distance, I in fact have to take this into account in order to make meaningful comparisons. Again, something that often is not done. So what about these highly derived model systems? So this is our August Krogh, a Danish physiologist who got the Nobel Prize in Physiology or Medicine in 1920 for work that he's done on vascularization, pretty important fundamental work. But he was also, as most physiologists at the time, a proponent of basically picking out different kinds of model systems, developing them, and asking interesting questions. And so what he wrote at some point towards the end of his career was, for a large number of problems in biology, there will be some animal of choice or a few such animals on which it can be most conveniently studied. And so I'm giving you a couple of examples here, such as we can learn a lot about animal communication and sensory processing of communicating signals from electric fish here on the bottom left. We've learned an incredible amount about auditory processing, especially spatial processing, where sound comes from in space, from work on barn owls, which are very good and highly specialized in doing that. And again, we've learned a lot about auditory communication from crickets, from the lowly cricket. Who would have thought? So, but of course the danger is that these creatures might be so specialized that whatever we learn, even though it is interesting, might not be generalizable. So we have to be very careful about this. So another problem that I've alluded to was already in some depth discussed by Stephen Jay Gould and Richard Lewontin in their 1979 paper about the Panglossian paradigm and the spandrels of San Marco in Venice. And so what architects discovered at some point is that they could construct buildings with fewer columns to support the ceiling and upper stories if they created these arches. And as a consequence of the arches with the columns, the space in between the arch and the column became available to do something with it. But it didn't have really an architectural or constructive function, right? It's just there, it's empty space. Now artists immediately seized that opportunity and said, we can do something with it. And that became so popular that architects then were forced to basically design spandrels in a particular way, sometimes even compromising the structural integrity of the buildings they were designing, because artists wanted to have space to do their art. And so, in a way, the spandrel has become very important. It has acquired a function in churches and many other kinds of buildings, even though originally the function was not there. So what the warning is here is that only because we see a structure in an animal, in a creature, doesn't mean that it does actually have an adaptive function. It may be a consequence of some other process, but later on it might become important and might become adaptive. Finally, is this issue of phylogenetic non-independence. So when people compare species, what they often do, if they are phylogenetically ignorant, which is unfortunately very often the case, is they assume that they all derived from a common ancestor pretty much at the same time. This is referred to as a star phylogeny. Now, there are groups of animals out there and groups of plants and other creatures that in fact follow our star phylogeny because a burst of diversification happened in a very short period of time, evolutionarily speaking, and they are all basically equally related. The distance from one species to another species is pretty much always the same. Now, most species, most animal species and plant species, however, are related in a much more complex and more hierarchical fashion, like what is shown on the right. And so that means if I want to compare different species, they may in fact be, the evolutionary distance between them might be different. And some may be more close related, some may be more distantly related, and we have to take this into account. Also, some may evolve more quickly, some may evolve more slowly, as indicated here with the different, how far they stick out from the top. And scientists over the last 25, 30 years have in fact developed a number of statistical models that are subsumed under the term phylogenetic comparative methods to deal with that, but we still need to educate a lot of biologists and the public at large that this is an important issue. Okay, so now let's go back to this basic idea that there are universals, molecular universals, underlying complex social behavior. And I want to be a little bit provocative here, because I'm the last speaker, it's right this time in the afternoon where most people would like to take a nap. And I'm tired too. I just flew in this around noon here, and I rushed over here to give you this talk. And tomorrow I have to teach, but I still have to make a lecture. So I need to keep myself thinking about this as well. So what does romantic love in humans and pair bonds in animals, what do they have in common? Well, remarkably enough, they have quite a lot in common. And one thing is that there's a dopaminergic system, meaning there's a system in the brain, a circuitry, if you will, that utilizes dopamine, which is a biogenic amine that is used as a neurotransmitter that activates neurons in a very similar fashion. And this is true not only across vertebrates, as they are shown here, but this is also true across all animals, including flies, including honeybees, including worms. And the other thing that I want to add to that is that this circuitry that is regulated by dopamine in all animals, especially in vertebrates, where it has been most studied, is also modulated on top of that, so which are smaller changes that might give rise to important differences in behavior by so-called neuropeptides, such as vasopressin and oxytocin. And chances are you've heard of oxytocin, especially if you're female, because oxytocin is very important for inducing parturition, childbirth. It's very important for inducing the milk letdown after birth. And it is also fundamental importance for the mother-child bonding. Without oxytocin, the mother will not establish an emotional bond with her child. And this is true, again, across many, many different species. So now what I'm proposing here is that, in fact, similar to the Hox genes, similar to the PAC6 gene that I talked to you about earlier, that the dopaminergic system, both molecularly and as we'll see in a moment, also in terms of the neural structure, is in fact a universal that is common to all animals, or at least to all vertebrates, and underlies very similar behavioral processing that we happen to call romantic love maybe in humans or the mother-child bond, but maybe if we would actually take away some of these terms that are highly loaded and fairly unscientific, we would realize that the underlying neural processes and molecular processes are fairly very similar to what we see in animals. So was that provocative enough? Okay. I'm obviously not a psychologist, I'm a biologist. So let's look at the dopamine system a little bit more. And so there are two levels of biological organization that I want to talk to you about. The first one has to do with the molecular and cellular level, where basically we have neurons that produce dopamine, and that is indicated here. I'm going to try it again. I'm brave. I hope that I won't blind anybody by pointing at you. So in this cell here, dopamine is produced. So dopamine is the neurotransmitter that allows the cell to communicate with its neighboring cell, with the postsynaptic cell. Is produced from amino acids such as tyrosine, and it is not encoded in a gene. It is produced from these building blocks that are floating around in the cell, in this case tyrosine. And then there are several enzymes. These enzymes are, of course, proteins that are encoded by genes. And a very important one is tyrosine hydroxylase, TH. And so tyrosine hydroxylase mediates the conversion of tyrosine to what we call L-DOPA. And then there's another enzyme, and eventually we get dopamine. And then the dopamine eventually gets released across the synaptic cleft here, and then induces a variety of cellular responses in the postsynaptic neuron. And it does so because there are receptors on the postsynaptic neurons that specifically bind dopamine. And that's why they're called dopamine receptors. So you find this in all animals. All of them do that. And remarkably enough, the functional context in which dopamine is used is also very similar, and we'll get to this in a moment, in terms of encoding the salience or the value of a stimulus, including a social stimulus. Now, we can exploit this very well understood biochemical pathway and visualize the dopaminergic system in the brain. And so in this particular example, what I'm showing you here is an in situ hybridization. This is from our own work, where we visualize the D1 receptor, which is shown here, the D1 receptor. So that allows us, by basically looking at the mRNA of the D1 receptor gene that is being expressed throughout the brain, this allows us to identify the areas in the brain that actually can listen to dopamine, that get an input from dopaminergic neurons. And then we can also, and this is another example of the kind of histology that we do in neuroanatomy, we can use tyrosine hydroxylase or an antibody that recognizes tyrosine hydroxylase to visualize dopaminergic neurons in the brain. And now we can see where these dopaminergic neurons project and whether these projections overlap with the presence of dopamine receptor. And there are different dopamine receptors, but we don't need to worry about this. Okay, so this allows us logically now to map out the dopaminergic system, not only maybe in mammals, but in all creatures if you want to do that, because this is conserved. This is basically 1 billion years old. Now, another striking discovery that was made just recently by Oliver Hobart's lab at Columbia University was that molecularly, the genes that are involved in specifying the identity of a dopamine neuron, such as tyrosine hydroxylase and some other proteins, or the genes that encode these proteins, they all share a common regulatory logic, meaning they have a motif in their regulatory region. As you probably know, each gene has a regulatory region that determines when and how and where it is expressed. And they have a shared motif here that is called a regulatory motif, where a transcription factor that has been identified, it's called ETV1, can bind. And this is true across all animals. Again, this system is 1 billion years old when animals first arose, multicellular animals first arose. And so you can take this particular motif and put it in the regulatory region of a mouse gene in a mouse from the worm or vice versa, and you will get dopaminergic neurons and functional dopaminergic neurons. So again, we have this deep homology, even though the structures of the nervous system are obviously very different between worm and mouse or mammal.
So from worm to mouse, but it's a
No, because, well, there is some variation here, but basically this is just like always, the transcription factor still binds. We have found very few things that make us unique, and I would just stop hoping for finding more. So we are not unique. And so the other thing that we now have to look at is, okay, we have a regulatory logic at the molecular level that indicates that dopamine signaling functions the same way across all animals. But then we also need to worry about the nervous system as a whole. As you probably know, the brain is a very complex organ. In fact, it's probably the most complex structure in this universe as far as we know. And chances are that as soon as we find out that there's something more complex, we'll be dead. So that means that now we have to start looking not only at the molecular level, but also at the neuroanatomical level, using these kinds of approaches that I just showed you with immunohistochemistry and in situ hybridization, basically doing good old-fashioned histology. And so what I want to show you now is actually the attempt to put on top of this these molecular universals of dopamine in dealing with dopamine neurotransmission, a layer of neural structure where we now use a framework that we've developed based on circuits, neural circuits that have been discovered and studied in mammals. And so we're going to do this across all vertebrates because that's how far we've gotten, and because it is a lot harder to extend in terms of neural circuitry outside vertebrates, and so I want to leave this for another day. So we look at the major vertebrate lineages, mammals, birds, reptiles, amphibians, as well as teleosts and other fishes, but our focus is on teleosts, and see, can we actually reconstruct these dopaminergic and neuropeptide systems across all these animals. And so the two mammalian circuits that we use to do that are first the so-called mammalian dopamine reward system. And it is called a reward system, somewhat misleading, which is why I put it in quotation marks, because this collection of brain structures that is schematically indicated here, this is the front of the brain, this is the top, this is the back, this is the bottom, because they are known and have been shown to encode the valence or the salience of a stimulus. And so, for example, in a learning paradigm where the animal learns a particular task and then is rewarded for it, this circuitry here becomes activated. And this is why originally it was called the reward circuitry. That's how it was first discovered. However, it is involved in encoding the salience of a stimulus in many other contexts as well, not only in a learning context. Okay? So, as a side, this circuitry is also the neural substrate underlying drug addiction. And so when you get addicted to drugs, the properties of this circuitry change, and basically, in order to have a rewarding experience, or to have some satisfaction, you need more and more dopamine, and you get that by taking drugs. And so you get this craving that you have to get more and more of it, and you can't get away of it. And that is commonly known as addiction. In that sense, and Larry Young, a colleague of mine at Emory University, he's done a lot of work on this system in the context of pair bonding and social affiliation in roles that you may have heard of. In that sense, because in social affiliation, when we affiliate with friends, or especially also when we fall in love, this circuitry gets activated in a way that is very similar to drug addiction. So falling in love is in fact an addiction, physiologically speaking. And most of us probably have experienced the withdrawal effects when you get dumped. So it's physiologically very, very similar. So the second circuitry that we use to build this framework to look for neural structure universals, at least across vertebrates that might underlie social behavior, is the mammalian social behavior network that was first proposed by Sarah Newman at Cornell in 1999. And so this is defined as candidate nodes for a neuroanatomical network that regulates sexual, aggressive, and parental behaviors in both sexes of mammals. And these are the structures down here. And again, I'm not going to go through them in detail, but suffice it to say that there's an overlap that there are structures shared between the reward circuitry and between the social behavior network. So what we decided to do is to take these two candidate circuits. You know about candidate genes. John Wallingford told you about candidate genes that he uses to explore biological questions. We now have two candidate circuits that we put together. They're already functionally overlapping, as we know, and neuroanatomically overlapping, and to compare across all vertebrates the presence and the involvement of dopamine and neuropeptides. So, but if we want to do that, we have a big problem. As you may know, vertebrate brains are incredibly diverse, and so this is from a textbook here, just indicating how different brains can look. And there are the weirdest kinds of brains out there. So, and they sometimes develop in different ways. And so the difficulty is to actually identify in a brain area in the brain the areas that are homologous, meaning that have similar function and that might be in a similar functional context where they are used. And a lot of work by comparative neuroanatomists has gone into that we're capitalizing on. But I want to highlight this problem that is a very important problem. This is a lot more easily solved, this problem, at the molecular level, because you can align nucleic acid sequences, DNA, for example, and because you have the dimensionality is so much lower, you have only 4 different letters and a limited number of combinations, it is a lot easier to identify these commonalities that are a result of common origin or homology. So here we have it much harder, but as it turns out, over the last 100 years or so, we've come a long way, and for the most part have not completely solved it, but to a large extent have solved it. So if we now look at these brains across vertebrates more closely, and so these are transverse sections, so if you were to section through the brain like this, from the front to the back, for teleost fishes, for amphibians, for reptiles, for birds and mammals, we've done this very carefully, and other people have done that as well. We can actually identify, based on lesion studies, based on developmental studies, based on looking for connections between the different brain areas, we can identify the homologous structures that are involved here, and they're color-coded here in all the different vertebrate groups. Okay, so now we can use histology, as I've shown you before, to actually delineate the dopaminergic reward system in all vertebrates, to delineate Newman's social behavior network, as well as the shared parts. And so we do that by doing neurochemistry, where we look at the neurochemical profiles, or the genes that are expressed that are important in neurotransmission and neuromodulation in these areas. And we already talked about tyrosine hydroxylase, which is an enzyme involved in the synthesis of dopamine, as well as the D1 receptor. We're looking at vasopressin and oxytocin receptor, as well as the peptide-producing machinery as well. And finally, we look at steroid receptors, sex steroid receptors, which obviously are very important for social behavior. I'm not going to go into those. So if we do that, and these are the different brain regions, if we do that, we find that overall there's a remarkable conservation across all vertebrates, and that is indicated to the right in this heat map, where basically each row here indicates a brain area expressing or not expressing a particular neurochemical that is listed here on the left. And then we simply clustered them by similarity. So orange would indicate present, blue or purple would indicate absent, and where it's white, there's missing data. So where our colleagues haven't really done their homework yet, and some of it we're filling in ourselves. And so one of the things that you realize, and that is pretty exciting to us, because as we'll learn in a moment, we actually work on fish. The mammals and teleosts are fairly similar in their neurochemical profiles in these brain areas. And so this does not actually reflect phylogenetic relatedness, right? Where birds and reptiles should be close together, which they are, and then there should be mammals next, and then amphibians, and teleosts should be way out there. But as you can see here from this cladogram, this is not the case. And so there are a number of possible explanations for this, and we can go into this later on if you're interested. But overall, there's remarkable conservation. Now, there are some exceptions here, such as the nucleus accumbens, which is important in processing the salience or the appetitive value of a social stimulus, shows some variation here, whereas other structures, like the preoptic area that we're going to go into in a moment, is almost totally conserved, and most likely this is an error in the literature, and we're trying to sort this out. So that basically for the past 450 million years, which was around the time when you and I and fish had the last common ancestor, this system was already in place for regulating, neurochemically in place and structurally in place for regulating social behavior. And so if we then take it all together and take a step back and look at the conservation or diversity, we see that there are particular brain areas such as the lateral septum and preoptic area, which is interesting for me, doesn't tell you much at this point, are very highly conserved, indicated here in red, whereas there are other areas that vary a little bit, but all of them overall are pretty conserved, so they are highlighted here. And then there are particular genes, neurochemicals, that are very highly conserved in their expression across all brain areas that are indicated down here. Coincidentally, that they have to do with the dopaminergic system. Okay, so now we've reconstructed basically a circuitry that is based on two circuits studied in mammals, the social behavior network and the reward circuitry, and have shown by neurochemical profiling that they are highly conserved across all vertebrates, and they are at least 450 million years old. And this is a summary picture that is of that ongoing work that shows you this. Okay, so I've given you two potential molecular universals that might be important in regulating behavior across vertebrates or even beyond. So the dopamine system on the one hand and neuropeptides on the other hand. But can we discover others? Are there others out there that might be important, and how would we do this? And this is where I want to show you some of that work that we do using genomic approaches. And so I want to give you two examples that really have to do with two of the most important decisions in life. There's a third one I'm not going to go into. But the first one is whether you should fight or not fight or run away. So what is actually, what are the molecular underpinnings of social dominance, especially in the context of male-male interaction? And while in our world it rarely escalates to this, luckily, the competition that we all experience in our social relationships in terms of the underlying neurobiology is no different. This is just it's a lot easier to determine here who's dominant and who's subordinate at the end of the fight than it might be, let's say, with your colleagues sitting in cubicles or so. And then the other example I give you is an evolutionary genomic analysis where we're going to consider the evolution of pair bonding and mating systems, and again use a genomic approach to see if we can actually discover some common molecular signatures associated with, say, monogamy or polygamy. So let's start with male-male competition first. Now, this is often referred to as intersexual selection because it is, as it turns out, in addition to female mate choice, which is across sexes, intersexual, male-male competition drives evolutionary change, eventually resulting in population differences and the appearance of novel species. And I give you a few examples here that are spectacular. I really like this one down here where two alligator males are fighting. And I'm sure that you don't want to get too close to that. And so we pursue these kinds of studies in a non-traditional model system, the African cichlid fishes. And we do that for a variety of reasons. If you've ever been to a pet store and instead of buying food for your dog, you want it off into the fish section, you've seen these fish because they're very popular in the hobby. It's quite clear why they are popular. For one thing, they're freshwater fish and they're easily raised and bred. Monkey can do that, as opposed to saltwater coral reef fish or so that are. Then they are very colorful and they are phenotypically, morphologically very diverse. But another thing that you don't see here is that they are incredibly socially sophisticated. They have very complex social systems. They have complex social interactions. They take care of their offspring, either only the mother or only the father or both parents together. In fact, in this group here, we find also several species of cooperatively breeding fish where they basically pool all their resources, even though they don't have to be related, and raise the offspring like in a hippie commune. So most of these fish that we study come from the East African Great Lakes, particularly Lake Tanganyika, Lake Victoria, and Lake Malawi, and they are evolutionary very recent. And the one species I want to focus on here is this one here, and I want you to memorize this because I will ask you later what the name of that species was. And in this species, the males come in two different flavors. The first flavor is a territorial male, very looking, brightly colored. He defends a territory, as the name already indicates, but he has something else that you can see here. He has nice big gonads, and he gets all the girls. And then the other flavor are these guys here. They are fairly dull in coloration. They don't have territories. They school with juveniles and females. And if they are threatened, they run away, and otherwise they don't do much. And they have tiny little gonads. So even if they wanted, they couldn't reproduce. So we call these guys machos, and we call these guys wimps. So we have machos and wimps. Now for those of you in the audience who are machos, listen carefully, because the macho very easily can become a wimp and vice versa. So this is like going into an undergrad student pub or so. It's very similar. So that what happens, some social change happens, the macho loses his territory, he becomes a wimp, or the wimp has an opportunity and becomes a macho. I'm going to show you a little video. I don't know when I started and how much time I still have, so you have to cut me off. Let's just look at the video here. This National Geographic did this a few years ago in our tanks. So here's the territorial male having sex with the female. And this is now a non-territorial male who's trying to become territorial. And so you see these ritualized displays that are very easily quantified. Well, this is slightly staged. That's the problem with popularizing science, right?
The new territory holder is defeated. He switches off his fancy colors. His gonads and brain region for sex will soon shrink. He rejoins the ranks of the wannabes.
Okay, so yeah, just let that be a warning when you go to the pub tonight. I've seen it happen. It's just not pretty. Okay, so now we're going to look at the preoptic area again. I told you already before that neurochemically it is highly conserved across all vertebrates. And it is now, I can tell you a little bit more about it, is a neuroendocrine integration center regulating behavior. And so where is it located? So this is a fish brain here. I give you all the different major structures. And down here, right where the optic nerves go into the brain, sits the preoptic area. That's how it got its name. It has nothing to do with vision. It's just the location. And we can go in, and this is a histological picture of it from the side. So we see these cell populations. And we can go in and section the brain and then use a laser to actually cut out the piece that we're interested from brain sections, the preoptic area in this particular case. And once we've done that, we can extract RNA and we can do molecular biology with it. So we don't get much, but we can do this quite reliably. And then what we can do is we can profile gene expression. Now we could do this again using a candidate gene approach, which we often do in a variety of study. But we can also do what some people call a fishing expedition, pun not intended, but what I would call a genome level study. And we can use a device that is called a microarray that we've built over the years where we can interrogate the activity of thousands of genes on a genomic scale. And so this is just a blow-up of some of these features on the array that represent different genes. And what we now do is, once we have extracted the RNA from the brain part that we're interested in, we do a reverse transcription to turn it into DNA, because DNA is a lot more stable than RNA, and then label the DNA with a fluorescent dye that is conveniently called green and red. And so then we can hybridize the cDNA to this array, and the relative proportions of hybridization to the different features that represent the different genes will tell us the relative expression or relative activity of the genes that we're looking at. So you get this Christmas tree pattern, and then doing a lot of sophisticated using laser optics and sophisticated statistics and bioinformatics, we can in fact then infer the activity of genes across the entire genome for this small part of the brain. So now if we do that, we can do a lot of things with it, but one trick is, when you deal with these large data sets, is visualization. Not only for me to communicate this to you, and you are not biologists by training maybe, but also to communicate it to my colleagues, and in fact to communicate it to myself to see what all the data might actually mean. And so one way to do this is to create this kind of a heat map. I hope you can see this, where each row here indicates a gene. We're looking at 630 or so genes at this point. And each column indicates a different individual, or the preoptic area from a different individual. And I'm just using 10 here to illustrate the point. And so then the color indicates whether a gene is very highly expressed, relatively speaking, or not. And so what you see here is that there are patterns, transcript patterns, that emerge, and that overall WIMPs, or non-territorial males, show patterns that are fairly similar to each other, and territorial males or machos do the same, and they are kind of in opposing direction, as may not be that surprising. So now we have a list of genes that we can visualize like this, but what else can we do with it? Well, I have 630 or 629 candidate genes now, and I could design a PhD thesis around each of them, and I could recruit 629 graduate students to work on that. That would be pretty crazy, and it would certainly drive me crazy, and I don't have the money to do that, so we want to do something a little bit more sophisticated. And so one thing that we can do is we can generate covariance matrix or network from these genes that if this is a gene here and this is a gene here, and the expression varies in the preoptic area across individuals, where each data point here would indicate an individual, we can do this for all these 630 genes, or even more or fewer, and construct a covariance matrix. And so, meaning indicating how tight is the correlation between gene I and gene J in this case, and we can do that for all of the genes. Then again, we have to visualize this, and one way to visualize this is again with this heat map here, and what you see is all these 629 genes are listed here across, and in the same order down here, and so you would expect that you get a diagonal, a very thin line diagonal, that where you correlate each gene onto itself, and so this would be an association or correlation significance that is very high. But what you in fact also see is a huge amount of variation, a huge amount of co-regulation, where there is this large chunk or cluster of genes here that appear to be regulated in the same fashion. There's a smaller cluster up here, there are some clusters here, a cluster down here, and another one here in the corner. And so there's a remarkable amount of statistical structure in these data that you wouldn't expect just by pure chance. Similarly, you can do this for non-territorial males, and you get a different picture. So now I've shown you a lot of pretty pictures. You can put them up in your bathroom, you can admire them, but is there any biological meaning in there? Here's a different way of representing this. You may have seen this because it's become popular in a field that is called systems biology. This is a gene network that we reconstructed from our data, where each node here indicates a gene, and each edge or line indicates the statistical association between this gene and its neighboring gene. And again, these clusters appear here that where the question now is, is there any biological meaning in it? And in fact, what we can do is now, since we've carefully quantified all kinds of phenotypic and physiological characteristics of these animals before we took their preoptic area, such as aggression, condition, stress response, sexual behavior, and so on, we find that in fact this large cluster here in the middle is strongly associated with aggression. The one up here in the corner is strongly associated with the stress response, and so on. So what we've identified now are co-regulated sets of genes, or modules, as some people call them, that appear to be associated and underlie differences in social behavior, such as territoriality and non-territoriality. We can do the same for non-territorial males. Now, the causal connection is not there yet, but we can, in fact, do perturbations of various kinds that I don't have time to go into to explore the causal relationship between behavior and gene expression in this brain area. Okay, and now we can annotate our network, and we find that this cluster here in the middle, for example, these genes are strongly associated with aggressive behavior, whereas the purple ones down here, this cluster is strongly associated with sexual behavior, and so on. So now we have biological meaning, and we've created a bunch of hypotheses that we can follow up with these functional tests that I've mentioned. Okay, so am I out of time now? Yeah, I can go on forever. I love this stuff. As John said earlier, if you are not exuberant about this, you're not going to do it. You know, I work, I don't know, 80 hours a week. I don't get paid much. You know, I deal with an insane administration, and I have to apply for grants. And then for every grant dollar that I get in, I have to give a chunk of money to the university. You have to love this, right? So, so we're all masochists. So I think I can stop here. This is just fine.
What about, we'll finish at 4:30, so leave a little time for questions.
Okay, so I can finish this up in probably 3.5 minutes, and then we have some time for questions. Okay? Thanks for your patience. Okay, so now let's look at mating system evolution. So let's go back, and I told you already that these cichlids in Africa cichlid fishes are incredibly diverse. Here you see a mother with the babies in her mouth. It's pretty cool. Here you see a mother that farms out her babies. We call that farming out. But when there is a sign of danger, she sucks them in like a vacuum cleaner. It's remarkable how fast it goes. In fact, she produces an auditory signal that tells the babies to go back into the mouth. Okay, so the species we're going to talk about now, they belong to a particular group that are called the Ectodini. I want you to memorize that as well. And within that group, we're going to focus on a few species that are listed here. And why is this group interesting? Well, first of all, they're all descended from a common ancestor here within the last 1,000,000 years or so. So this is rapid evolution. Things go pretty fast down in Africa with these fish. The other thing I want you to realize is that some of these fish are monogamous and some of them are polygamous. Monogamous are green, polygamous, whereas we say polygynous, are red. And the third thing I want you to notice is that the monogamous species are kind of distributed across this entire tree. So indicating that, in fact, there have been multiple independent transitions from the ancestral state, the ancestral mating system, polygamy, to monogamy. Now, this is a beautiful model system now for us to explore whether the same or different gene sets get recruited repeatedly in these independent transitions. And of course, to ask what kind of genes are involved in monogamy versus polygamy in the first place. And so how could these transitions happen? Well, behavioral ecologists and evolutionary biologists have pretty much figured out what drives mating system evolution at the level of ultimate causes. And so mating system evolution is often driven by biases in sex ratios. So if there are a lot of males but very few females in the population, if a guy finds a female and can mate with her, he better stick with her, because sooner or later she can mate again, instead of wandering off and trying to find another one because there are just not enough females available. The resources may differ, right? So if there's a lot of food available, then the male or the female, depending on the species, may say, well, my partner here can take care of the animals by himself or by herself. I'm going to go off and find another partner and increase my reproductive fitness this way. I'm anthropomorphizing here, obviously, but I'm trying to communicate these concepts that are actually fairly complex. Or predation pressure could also drive this. So, for example, if there's a lot of predation, if it's really dangerous for your offspring, it might actually be beneficial for you to stick with your partner and to help raise the offspring. Whereas if there is very low predation and it's very unlikely that your offspring get eaten by some nasty predator, you might as well take off and find another mate, right? And later you can ask me whether humans are monogamous or polygamous. Okay. So the question we ask is, have the same sets of genes been recruited repeatedly in these convergent or parallel transitions from polygamy to monogamy? And so one approach to do this sounds terrible. We have to do field work in Africa. So this is a picture of Lake Tanganyika. It's beautiful. Our site is really beautiful. Once you get there, it takes about a week to get there. And there's not much infrastructure, so we have to bring everything. And then this is just proof that I've actually been there, and some of my students and collaborators. And so one of the first things we did is actually we took brains out of these animals and we quantified them. And this, the brain structure, and to make a very long story short, one of the things that we found is that the size of the forebrain varies with social organization. And so we haven't done all the species yet because they're not all that easily found. But what you see here is that the monogamous species that we've sampled so far, and this is data only for the males, have a larger telencephalon or forebrain compared to the polygamous species. Now, if you are a defender of, family values, then you will say, yeah, this makes sense. Because, of course, if you're monogamous, if you're faithful, you have a larger brain. Now, if the result would have been the other way around, I could tell an equally convincing story. And that is the problem with brain evolution studies. They're all just so stories. So we did this in order to, again, identify candidate brain regions that might vary across species in association with mating system, so that we then can go in and probe them using this approach that I showed you earlier, using a laser microdissection and to look at the molecular signatures. So for us, this is just a means to an end. Okay, so now we actually pick 4 of these species that represent 2 of these independent transitions. There's a transition here, and there's another transition here. They're a little bit further apart, but when we chose those species, we didn't have the final tree yet, so that's the risk of doing science. And then, this is now color-coded differently. We are now comparing the transcriptomes of these species, and what you see is we find about 4,000 genes, about a quarter of the genes that we are analyzing to be differentially regulated. This is a huge chunk of genes, right? But we find that most of them, close to 3,000, are in fact associated with lineage. And so Leptura, these 2 species, and Tenioidinata, they are closely related, and in terms of their gene expression profiles, they're very similar. And Flavipinis and Ochrogenes are very closely related, and their gene expression profiles are very similar. So it doesn't look like it has anything to do with mating system, right? And this is what I said earlier. This is the danger of doing comparisons, because these obviously have a much longer common evolutionary history, and so it's not surprising that the evolutionary history, or as we say, lineage, will dominate the comparison. But if we look a little closer, we actually identify about 1,200 genes that in fact vary with mating system, that are upregulated, say, in the monogamous species and downregulated in the polygamous species, or vice versa. And so now, to make a long story short, we can in fact identify a couple of thousands of genes that are associated with mating system. And when we then look at the individual animals to see how they fall out in their expression profiles for these genes, we find that in the females are all over the place for these different species. The females can vary a lot for these genes in their activity, whereas the males are fairly cleanly, not 100%, but fairly cleanly cluster in that the polygamous species, polygamous ones are out here, and the monogamous ones are actually fairly similar to each other. So which indicates that the evolutionary transition was in fact driven by the males that went basically from being polygynous to become monogamous. And in order to do so, from a gene transcription point of view, they had to constrain themselves. They couldn't vary as much in order to make this transition possible. And so Well, what this means, if anything, I mean, these are obviously ongoing study that I'm telling you about, is that we actually now have data in hand where we can look not only at this systems level, but at individual gene level, which genes are actually associated with these different mating systems. And again, all this work wouldn't be possible without a bunch of people in the lab, graduate students, postdoc, research assistants, undergraduate students, and of course, there also needs to be money that usually comes from various sources. And with this, thank you very much, and I'd be happy to answer questions.
Is there anyone who'd like to come up to the mic, or I can bring it to you? And in the meantime, could you answer your own question that you told us to ask you about humans?
I think it is a nonsensical question. So there's pretty good evidence if you look at the structure of our genitalia that we are probably polyandrous, that females are evolutionary speaking promiscuous, and that males are just scrambling to get a piece of the pie. But of course we could argue for this over a beer for a long time. But both the human vagina and the human penis are very unusual compared to our closest relatives, and so some interesting arguments have been made about that.
Could you say something about the time it takes for that transition from territorial to non-territorial? What's the gene expression profile in that case?
Yeah, we're currently exploring this with a very nifty little experiment where we're looking at the transition from wimp to macho. Behaviorally, the transition goes like this. So we have a tank that is where we have an animal that is basically forced to be a wimp, and then one morning my student Lynn goes into the fish room before the lights come on, with a red light on her head because these fish cannot see red, so it's still dark for them. And she yanks out the territorial or dominant male. We call this a bullyectomy. And then 10 minutes later, the lights come on, and the wimp, for the first time, doesn't get harassed. And within 15 minutes, he's as aggressive or even more aggressive than a territorial male. His androgen levels, such as testosterone, go up incredibly rapidly, at least within 30 minutes. And he will be but his reproductive behavior and reproductive system, in fact, takes longer, about 5 to 7 days. And after 2 weeks, we consider him to be a fully established territorial male. So on the way down, the behavior shuts down right away, but physiologically, they hold on to reproductive physiology undercover, so to speak, for up to 2 to 3 weeks.
What about the, oh, sorry.
What prompts the conquest? When is that choice made?
So a male can only reproduce if he has a territory. And the territories are shelters such as banana leaves or rocks or plants, and it's a limiting resource. There are more males than there are territories. And so a non-territorial male may eventually be in a position in terms of size and condition where he can challenge a territorial male. And then the territorial male may lose a fight, just as you saw in the video, and then the other guy takes over.
What prompts him to turn tail, if pardon the expression?
Pardon me?
What prompts him to turn tail? At what point in that fight, that clash, does that happen?
That is $1 million question. A lot of people have thought about this.
You talked about conservation with that particular transition, which is so dramatic in the fish that you're studying. What other examples do you see of that transition in other species?
Oh, in other species. Well, I mean, think about life history transitions in our own species, right? Think about puberty. Think about as we go through different life stages, as we become older, as we have children, as the children leave the house, as we become grandparents. So from a life history perspective, these transitions are not that different. They may not be as dramatic as what we see here. At least many of us don't perceive them as dramatic. But for a Martian, they may in fact be just as dramatic. The other transitions is when we move, when we change jobs, when we get into a different social circle, we actually have to establish ourselves within that circle. Now, it's not that we start fighting with people, at least most of us don't, but again, we have interactions and we have to figure out where we fit in and whether we have going to have a higher status or a lower status. And what's interesting about modern humans is, because we move in so many different circles, is that we can actually play different roles in different contexts fairly well. So I would just leave it with humans, but there are many other examples. Yes, please.
Okay, last question. Judy.
That humans are likely to be polyandrous, that the structure, the behavior, or whatever of the sexual organs suggests that females are prone to be promiscuous and males just have to scramble. I wondered if you could elaborate on that. Thank you.
I'm not sure I want to do that with this audience, because you guys are going to quote me on that and I'm going to be in trouble. So the human penis compared to the penis of other hominids, other primates such as chimps and gorillas have tiny little penises, right, like an inch or so. Now chimp males have large testes. They're stock full with sperm because it's a polygamous mating system where the male tries to get as many females as possible and he doesn't want to run out of sperm. That's different in gorillas that have very small testes and in fact have a different mating system. Now the human penis is fairly long, but and so it was long thought that it is females that have a preference for a longer penis. But when you actually ask women about the preferred length of the penis, they end up with some sort of intermediate size, what their preference is. And so this seems to be true across cultures. And we can argue about how reliable these studies are and so on. I'm just giving you the kind of story here that seems to make sense at this point in time. Now, so then the question was why could, and also the human penis has some strange features, such as the glands and so on, that actually can create a certain suction movement, which is thought, and experiments have been done, believe it or not, that it can actually suck out the sperm of the guy who inseminated that female before, right? And so that is, for a male, this is of course great. If you have a promiscuous female, you pull out the sperm of the other guy before you inseminate that female yourself. Another thing, the human penis has this ridge, I forget what it is called exactly, that is thought to stimulate the chi point, and you don't find that in other primates. And the chi point, supposedly, again, the literature is very weird on this, makes it more likely for females to actually have an orgasm, and a female that has an orgasm during sexual intercourse is more likely to ovulate. And if she ovulates, she's more likely to conceive from that male that she has intercourse with. So that's kind of the story, and just don't attach my name to it. I mean, a lot of, it's plausible.
Earlier today, some of the tweeters were saying, I really have a hard time getting some of this in a tweet. So I challenge any of you who are trying to summarize this to do so. Let's thank Dr. Hofmann for joining us this afternoon.
Thank you.
He's as tired as many of us are because we've been on our own marathon for